Welcome to Derms on Drugs, a video podcast brought to you by Scholars in Medicine, the best educational platform in dermatology, and provided at no cost to medical providers. Derms on Drugs is where cutting-edge derm meets hit-or-miss comedy. I'm Matt Zires, and each week I'm joined by my residency buddies, Dr. Laura Ferris and Dr. Tim Patton, to use our 60 years of combined derm experience to discuss, debate, and dissect the hottest topics in dermatology. It's everything you need to know to be on the cutting edge of derm, and you'll actually have fun listening. New episodes drop every Friday on Scholars in Medicine, Apple Podcasts, Spotify, and other major podcast platforms. Just a note for everybody, the video component has the key figures and tables from the articles we talk about, and the episode description has a link that'll get you to the links to the articles and other resources that we discuss on the episode. So this week we've got one of our patented six-pack episodes where we are going to go through our 6 favorite articles from the recent literature, and we are going to jump right in with Dr. Ferris. Dr. Ferris, what do you got?
Okay, Matt. So my first article was in the Journal of Cosmetic Dermatology, Zoe Drelos et al. And this is a 12-week randomized, double-blind, placebo-controlled trial for the efficacy and safety of a novel nutraceutical for mild to moderate acne. What is that nutraceutical? It is Nutrafol Acne. Okay, what is a nutraceutical? These are basically things that are like botanicals, but it doesn't meet the criteria to be a drug, so it goes through a different approval process. But this is basically a combination of a bunch of things that address the root cause of acne. I think we all get tired of hearing the words root cause from patients, but that is what they do. So root cause stress, you address that with holy basil, diet.
Holy basil.
What's that?
Nothing. I love that name.
Holy basil. And our, in honor of our new pope, we've got holy
Not
basil.
sinful basil,
No.
holy basil.
Holy basil.
Okay, all right.
Berberine for diet, metabolism, hormones, maca, HP, and selenium. The microbiome, the couple pro and postbiotics, oxidative stress, some Sicilian orange, lycopene, olive extract, ginger extract, and then the immune response, curcumin. So it gets at the root cause of acne. Okay, so this is a 12-week randomized, double-blind, placebo-controlled study, which you don't always see with nutraceuticals. This was only done in women who had mild to moderate acne. They were washed out of all their other acne products like topical retinoids, and they were put on a standardized skin regimen, which was basically Cetaphil cleanser, Cetaphil lotion, and Neutrogena sunscreen. And they were given that. And then they had the option of using a rescue medicine, which was adapalene 0.1%, but nobody did it. Okay, so what were the inclusion criteria? They had to be women 18 to 50, mild to moderate acne, like 5 inflammatory lesions, 10 non-inflammatory lesions, and an IGA of 2 to 3. So this is a score of 0 to clear to 4 severe. So 2 is mild, 3 is moderate. So at baseline, the groups were pretty matched. They had an average IGA of 2.2 and 2.3. And then if you look at, then the patients were given their medication at week 12. The mean IGA for the active group went from 2.2 to 1.47. And in placebo, it went from 2.33 to 1.98. This was a statistically significant difference. The percentage of subjects that were clear or almost clear, meaning they had an IGA of 0 or 1, was greater in the active group by week 8 and continued to increase to week 12. So it was 44% in the active versus 13% in the placebo group were clear or almost clear. The percentage of subjects improving over the course of the study was also significantly greater. What did not differ, which was kind of interesting, was lesion count at week 12 between the two groups. Now, if you look at the graph, you'd be like, wait, that looks like a statistically significant number. But what they found was that the p-value was showing that the lesion count was statistically significantly different at week 12 than it was at baseline, but the two groups did not differ between each other.
So wait. The two little asterisks that they have are just saying it is statistically significant from baseline,
Yeah.
but not to compared to each other.
Not compared to each
A
other.
lot of acne studies do that. They have the two asterisks, and you're like, okay, yeah, statistical significance, but they're comparing it to the baseline, not to each other.
Right.
Bastards.
Yeah.
Bastards.
Yep.
Okay.
So they
Which is
looked at
weird,
some
right?
other things like, what's that?
How do you get great, like how do you get a much better score to getting to clear or whatever, almost clear, when there's no difference in lesion count? That didn't make any sense to me.
Yeah, I have a hard time, and I don't do acne studies, so I'm not super well calibrated on, how that would be. But it would seem, I mean, maybe there's like, so one of the things that they did do is they had readers basically look at photos, and then they looked to say, what percentage of subject achieved a 0 or 1 in different parameters. So, post-inflammatory erythema, hyperpigmentation, scarring, tone, smoothness, softness, overall, so they did like, it did seem like, there was probably a difference in things like hyperpigmentation, erythema, scarring may have looked better. So I guess you could say that there was an improvement. So I thought that was kind of, that is maybe like if you're doing a global assessment, you're not basing it on the number of lesions, you're basing it on all these other factors as well.
Okay. I mean, this is a great drug, right? I can't wait to tell the patients that are like, I don't want to do antibiotics.
Yeah.
I don't want to do antibiotics. I don't want to take anything that's going to mess up my hormones. Isotretinoin is going to give me depression. Please help me. I'll be like, I have your answer.
Yeah, I went and I looked up at I was like, let me look at studies of like Cabtrio and Winlevi to see what they looked like. And so, if they look at like treatment success, they'll find like 50 for Cabtrio, it's about 50% in the Cabtrio group versus about 25%, 20 to 25% in the vehicle group. Inflammatory lesion reduction, they did see like a significant difference. It was like 75 to 80% reduction with Cabtrio versus about a 60% reduction in inflammatory lesion count with vehicle. And so, they did, and then Winlevi was IGA success, so clear, almost clear, sort of the same endpoint. It was 18% in the Winlevi group versus about 9% in the vehicle group.
Now, were they enrolling mild, moderate in those trials, or were
No,
they enrolling
these were
moderate,
to severe. So it is a difference.
A harder endpoint to get right, because the 2-point improvement is, as a challenge.
So IGA success, Cabtrio about 50%, Winlevi 18 to 20%, but there's a difference here. So, I agree. Like, I like the idea that like this is not an antibiotic. There are a lot of patients who don't want an antibiotic. They want something natural. They want to get at the root cause of their acne. Gosh darn it. And I can like now say that this will do that. It is out of pocket about $80 a month.
Yeah, they don't need our prescription. They don't need refill. I can't wait to start telling patients about that. Only if they like refuse isotretinoin. Don't get me wrong. I'm still giving isotretinoin.
And again, this is not isotretinoin level patients. So yeah.
Oh, I think mild acne totally deserves isotretinoin.
Yeah, that's when my daughter got like 4 pimples at once. Isotretinoin.
Yep.
It's crazy to me how many. I really believe there's a huge nocebo effect with isotretinoin. The reason that anybody gets depressed on it is because they read they could get depressed on it, and then if they just naturally normally get depressed, they blame it on the isotretinoin.
Yeah, I agree.
I think it's so you guys are both gonna, so $80. See, $80 seems, says $1,000 a year, but I guess if you've got acne and you're one of those 44% who get to clear, almost clear. So I guess the thing to tell people is, hey, go on this. There's a 50-50 chance it's going to be amazing for you. And after 3 months, if you're not doing amazing, it's probably not worth it. But you spent $250 to find out. I feel like it'd be smart of the company to give like 3-month starter packs. or something like that. Let people find out if it was going to work or not, because that $80 is a pretty big sticker price.
Yeah, although sometimes co-pays are that much. And, also like this may work in combination with some, topical retinoid, right? I mean, so this is monotherapy. Who knows what it's going to do in addition to, benzoyl peroxide, tretinoin, and clindamycin, right? So I think it'll be interesting. I like having it as an option.
Yep.
What I've been doing a little more of lately is, especially for postmenopausal women or for guys, is just very low dose Accutane almost indefinitely. So like 10 milligrams 3 times a week, because you don't really get any dry anything. And it's actually really, really effective. It's just you can't do it for women of childbearing potential because it's too hard. It's too much. There is a real risk that people are going to accidentally get pregnant if you have them on it for years. But the low dose long term for isotretinoin has been really good.
Yep.
All right, let's move on to our next article. So again, that was just before I forget, because we're not sponsored by anybody, so I don't want anybody to think this is a plug. But that was for the Nutrafol
Acne.
Acne. And can people get it on Amazon, or do they have to like buy it in a derm office? Like, do you have to sell
You
this
can
out of
buy
your
it
office?
online. Yeah, I looked it up. You can buy it online. I
Go
don't
to
know
the
if
website.
Amazon specifically, but
Okay.
But straight from Nutrafol. Yeah.
Yeah, I'm gonna get the counterfeit stuff from Amazon that's got lead in it.
Good.
But it'll only be
plan.
$65 a month for an ineffective toxin, so it'll be fine.
Good.
All right, Patton, what do you got?
All right, from the JAD, available in April 2025, titled "Skincare Ingredients Recommended by Cosmetic Dermatologists: A Delphi Consensus Study" by Gabriela V. Alvarez et al.
I gotta say, I've been excited ever since you picked this article to hear what you're gonna say about it.
Absolutely love this paper. Like, I love it. I will reference this probably more than any other thing that I've read in the last 5 years. So what did they do? Literature review from 1990 to 2020 generated a list of 318 ingredients, and that got whittled down to 89 ingredients by one group of experts. And then those 89 ingredients were evaluated by a second group of experts, and it went through 2 rounds of a Delphi panel made up of cosmetic dermatology experts. If 70% of the experts gave an ingredient a ranking of 7 to 9, 1 being the worst, 9 being the best, and no more than 15% of the experts gave it a ranking of 1 to 3, ingredient was said to have reached consensus for the condition being studied. So the final answer is in Table 2, a list of ingredients that we can now tell all our patients experts recommend for fine lines and wrinkles, acne, redness, dark spots, large pores, dry skin, oily skin. Table 3 goes through the consensus ingredients and lists the evidence supporting their use. So there's some science here. I didn't, let me see. Yeah, so, these are the ingredients, right? So for fine lines and wrinkles, it's mineral sunscreens, retinoids, vitamin C, chemical sunscreens. For redness, mineral sunscreens, sulfacetamide, niacinamide, green tinted products, metro, bromonidine, ivermectin, right? And when patients are like, well, what about for large pores? Experts say retinoids. That's it. They looked at other stuff, but they're not recommending it. It's a really, really Table 2. I'm going to carry this around with me every day, and I will bring it out all the time. And they did look at things like growth hormones and peptides and hyaluronic acid, and they didn't make the cut. So we can tell those patients, yeah, don't worry about that stuff. All these experts said that is advertisements and a bunch of crap. Clascoterone, talking about Winlevi, didn't make the cut. But I think that things like this highlights what's often discouraging about being a dermatologist, right? If somebody comes in and says they've tried their sister's Winlevi and they absolutely loved it and it works so much better than anything else they ever got, like, I'm not going to say, hey, yeah, experts said it was crap, I'm not prescribing it. I tell them it's great. I send in a script. And I don't think that happens with like real doctors. Like, no one goes to their oncologist saying, I tried some of my sister's dacarbazine and it worked way better than your boy. And one of the things from personal experience, like, I use mineral oil for my dry skin. I just put it on right after I shower. And, the expert said, yeah, mineral oil, that didn't make the cut. But So, like, are they just making stuff up? Like cysteamine hydrochloride, that has good data for treating melasma. Did it not make the cut because experts didn't like it? I don't know. I don't know what to make of this. Like, I don't know how real this expert opinion is, but it's just going to be very helpful because I can now show patients and say, here's what experts say.
So see, I think cysteamine has had good evidence for, and what I liked about this article was it's not evidence, because so many of these ingredients we don't have like great, because no one's going to spend, $20 million to do a study for, a topical that cost $9. But so what we're getting here is really when you look at the list of authors, it's like a lot of the heavy hitters in the aesthetic space. And so I think it's a blend of like, what have they seen in their patients?
Well, they went through like what reached final consensus. They went through what evidence was available, and most of it had like 1B, 2B evidence. It's not like there's none. Studies have been done, 1B, like the really good studies, 2B, smaller, but like still a study. And then the two things that had actually no evidence was the green tinted like cream to reduce erythema and benzoyl peroxide to treat oily skin. So experts still recommended it with very little evidence. Most of the other stuff actually had good evidence. But right, some of the stuff they said was not good also has good evidence. So I'm curious, like, what about that makes them say we didn't recommend it? I don't know.
I think it's their experience.
Yeah.
I don't know. Ferris, I hadn't thought of using this list that way. I am gonna use it now because this is all stuff you can give to a patient and say, go find a good product on Amazon, find something or online, look for stuff with good reviews and whatever, make sure you're getting the real stuff. But yeah, this does make a ton of sense. I hadn't thought about how useful it could be.
Yeah, and the newer stuff, the growth hormones, the peptides, hyaluronic acid. I think it'll be great to be like, yeah, as of now, experts like looked at that. They said no.
I don't know. From Dr. Ted Lane, I'm now a big believer in exosomes.
He was not included, and that is kind of disappointing.
That is. They were probably intimidated. Didn't want to bring on the real expert.
He's the Derms on drugs like cosmetic guy.
Yeah,
he should have been included. I don't know if I trust anything anyone else would say.
Yeah, I would agree. Yeah. Ferris, you got anything to add about this?
No, I agree. It's just nice to be able to say, like, the bane of my existence is like, what do I do about large pores? I've adopted the phrase, I'm a doctor who treats disease. I'm not somebody who does skincare, but now I can say, if you want data, here's what it is, but I'm not going to do a prior auth.
Go get a retinoid.
Yeah.
Go get some OTC adapalene.
Exactly.
Little pearl for everybody that I actually really like. If you ever get a patient who's asking us about like wrinkles and whatever on their arms and their body and the whatever, retinoids work. They have some effect even at very low concentrations. I will have people get a 30-gram tube of OTC adapalene, squeeze it into a jar of usually CeraVe, but it could be Cetaphil or whatever, mix it up to make a 0.01% adapalene. I don't know if it actually does anything, but I think it probably would if you did it forever. But it's, it's cheap and easy. That's what I like. Cheap and easy. Right. That's right. All right, let's go on to my studies. So I've got two that we're going to do back to back, and this is going to be a topic that I think we're all starting to see more and more about because Leo has a drug coming, delgocitinib, and so they're starting to do a lot of disease state awareness messaging for hand eczema. And so we had a couple of really useful things that just came out. So the first one that I want to talk about is the head-to-head trial of topical delgocitinib, which we're expecting to get approved here in the next couple of months in the U.S., already approved in Europe. So topical delgocitinib cream versus oral alitretinoin capsules in adults with severe chronic hand eczema. Delta Force, so named it after a cheesy movie from the 90s, I believe. A 24-week randomized head-to-head phase 3 trial. And so essentially people had to have severe hand eczema, which like when you look at the way that the IgA was developed, like there are people that we would say had severe hand eczema. could be any. It wasn't like, oh, you had to get patch tested and get contact derm. Like if you had hand eczema and it was severe, you could be in the study. People got randomized 1-to-1 to either delgocitinib cream or alitretinoin capsules, and the alitretinoin is a systemic retinoid. Side effect profile not that different from, say, psoriatane. Depends on the dosing, how likely you are to get dry skin and that kind of stuff. But alitretinoin, 30 milligrams once daily, which could be reduced to 10 milligrams a day. And really what it showed, the Delgo was substantially better than the approved oral drug. So alitretinoin is approved in the EU and Canada. It's the only drug out there that's been approved over the years for chronic hand eczema. Now they've got Delgo as well. But it was clearly better, like meaningfully better. Change in Hexi. So at baseline, the average Hexi score, and Hexi is Hand Eczema Severity Index. You could think of it, same general concept as a PASI or an EASI score. So it's a 0 to 360 scale, but severe usually is considered to start at about 70. And so the mean Hexi at baseline was 92. And of the delgocitinib treated patients, a significant number, so about 40%, 39% to be exact, got to Hexi 90 at 3 months, as opposed to 26% of the alitretinoin patients. So about 50% more people got to, in fact, exactly 50% more people got to Hexi 90 at 3 months, which is a big deal. It was interesting in the study, patients could, it was a really kind of cool design. Once people got better, the investigator could stop the drug. And then if the disease came back, they would restart it. About 28% of Delgo patients got so good that they stopped the drug. 20% of alitretinoin patients, same thing. Both groups, about 60% of the patients restarted at some point. But that explains why in the figure showing Hexi 90 proportion, you saw the Delgo have like a drop from week 16 to 20, and that was because at week 16 is when they could stop some people, and then they stopped them, and so they lost Hexi 20 or Hexi 90, then they restarted and regained it. But so, useful data, 40% of people did spectacularly well. They didn't really give data that on, say, in the main article on, say, Hexi 50, which is sort of the, like, in my mind, the drug's like helpful. But good numbers. And then we got a second article. So for our listeners, you know that here on Derms on Drugs, we love oral roflumilast, basically a free drug. So about $6 a month through Mark Cuban's Cost Plus Pharmacy. And think of it as a Premolast that is more potent, but exact same adverse event profile, GI side effects, but not maybe depression, but no immunosuppression. So in this study, it was an open label, 14 patients with chronic hand eczema, mean baseline Hexi of 86. So these people had, hand eczema that was comparably bad to the delgocitinib alitretinoin trial. Of those 14 patients, 13 got to 3 months. So only one of the 14 dropped out in those first series of months, and that was due to the adverse events. And the vast majority of the patients had GI adverse events at some point. So I think 4 didn't have any GI side effects, but 75% of them had some GI side effects, which I think is a useful number to keep in mind. And so of them, though, at 3 months, so pretty comparable to the study we were just talking about 29% got to Hexi 90. So 4 out of 14. So we're doing an intent to treat analysis. So the person who dropped out, we're still counting them. So 4 out of 14 got to Hexi 90. 9 patients made it to 9 months. 8 of those patients got to Hexi 50. So over half of patients, so 8 out of the original 14, it at least got them what I would call very meaningfully better. And the nice part about this drug, once Delgo comes out for hand eczema, or to a significant extent, you could call a lot of hand eczema atopic derm, and you could use Vtama or Opzelura or Zoryve if you wanted to. But so, Delgo is going to be the only one that has an FDA approval for chronic hand eczema of all types. You can use roflumilast together with Delgo, and I think it's going to be, they're mechanistically, there's so little overlap. I actually think we're going to have super effective treatment for the vast majority of patients with hand eczema, assuming we're able to get the Delgo covered. Really nice article, give us an idea that this is probably in the same ballpark of efficacy as Delgo. But so now we've got a good oral that'll be available in the U.S. that's very safe and easy to use. Not terribly effective, but safe and easy to use and cheap. And we'll have a highly effective topical that we can pair with it. Any thoughts or comments from you guys?
No, I thought it was interesting, and it's going to be good to have options for these patients. I oftentimes use like acitretin. As long as it's not totally eczematous, like if it's like hyperkeratotic. But yeah, I think it's interesting, and more options is always good.
Yeah, it's Patton. What are any thoughts?
No, why doesn't Delgo go up against steroids? That's what I would want to see, because I think if this is anything by like topical ruxolitinib, it's just going to be hard to get. And am I just going to be like, just steroids might be just as good?
Well, it'll certainly be, for hand eczema in particular. I think everybody will give a trial of clobetasol first, and then if it doesn't work, we'll go to Delgo. The challenge with hand eczema is that every case of hand eczema has a component of irritant derm. While clobetasol helps with the inflammatory component, it makes the underlying irritant derm worse by impairing barrier formation. So it'll be interesting, right? Once we get to play with it, because nobody in the U.S. has used any Delgo. The trials were completely done in the EU. So I have no sense of like, how does this compare to say Opzelura or Vtama or Zoryve? Just don't know. So it'll be interesting to see.
Yeah.
All right, let's move on to our next article. We're back. Speaking of Zoryve, we're back over to Dr. Ferris. What do you got?
We're back on roflumilast, this time talking about the foam, roflumilast foam, 0.3% for psoriasis of the scalp and body, the Erector phase 3 randomized clinical trial, Melinda Gooderham et al., just published in JAMA Dermatology. It's a phase 3 double-blind vehicle-controlled randomized clinical trial. So patients were 12 and older, plaque psoriasis affecting up to 25% of the scalp and body, but they had to have at least 10% of the scalp involved. And they also had to have non-scalp involvement as well. So they had to have.
It wasn't an either or. It was they had to have both scalp and body.
No, sorry, I said that wrong. So they had they did have to have scalp and body. So they had up to 25% total. They had to have at least 10% of the scalp involved and up to 20% of non-scalp involved. Sorry.
Okay. All right.
They also had to have a scalp IgA of at least 3, which is moderate. So they had to have, significant scalp disease. This wasn't just like a little bit of flaking. And then a minimum body IgA of 2, which is mild. Co-primary endpoints were scalp IgA and body IgA success, which is clear or almost clear with a 2-point improvement, so 0 or 1. And this was this was at week 8. Okay, so of 432 patients at week 8. Basically 2/3, 66.4% of the roflumilast group achieved a scalp IgA success versus 27% in the vehicle. And in terms of body IgA, it was 45.5% of roflumilast versus 20% of the vehicle. So I thought this was really interesting. One, foam is something that sometimes patients really like. 2, the efficacy in the scalp was really good. And it was better than what we saw on the body. Other things that were.
So how is that possible, Ferris? Because the scalp, normally the scalp is not like you think of scalp as a reason to do systemic therapy because topicals don't work on the scalp. So how
I think scalp psoriasis is a different entity and that it overlaps with sort of seborrhea versus body psoriasis. I just think psoriasis on the scalp is different. I think it behaves a little bit differently. I think it is probably driven by other factors like, the scalp oil is very different than the sort of microbiome of the body. I
Yeah.
think that it responds differently. I don't know. Patton, what do you think?
I know. Did the patients have to have hair? Like, did that matter? Like, if
No.
you're a bald, is your response just like the skin? Like, is there something the hair mixed with the Zoryve is doing?
So most people had hair, is my
Okay.
assumption. And if you look at their photos, most people, I mean,
There's
there's
only
a ton of
there's
photos.
only 2 pictures though.
There's only two pictures. Yeah.
Yeah.
My guess is this was not a study full of bald men.
Patton, did you get invited?
Even you
Hey,
have long hair.
I got pretty good coverage.
Yeah.
So wait, I talked to the company about this, and I have a theory
Okay.
for why it's better on the scalp. And my theory is this: the vehicle that they used is Crotofos, and the roflumilast itself. They're negatively charged, and your hair is negatively charged. And so, right, if you think about like here's a hair, if you put a drop of this foam on there, it should spread. It's trying to get off the hair. And so it's being repelled by the hair, so it spreads in both directions. Well, if you're putting it on near the scalp, so we'll say here, as it spreads in both directions, it's actually pushing it down into the follicle. And so I think, and this is totally made up.
So my hair thing, the hair versus bald that's a good.
I think you're right on. I think that it uses the hair as a delivery mechanism.
Okay.
That is my use for roflumilast foam, because for sebderm, I think I did the trials. 93% of people had scalp disease, so it was hard to like separate out in the trials. But my experience is, I think it's a really good for sebderm on the face and other places. Miracle for sebderm on the scalp, and that was what got me thinking about this originally. It's like, how can it work so well on the scalp? That is my made up answer: is that it's using the hair shaft as a delivery mechanism.
All right.
Yeah.
Is that a different, like Olux foam does not have that super
No,
repellent?
they are like very proud of their vehicle. It has this like,
I've seen that.
yes. I can't explain the science of it super well.
I
but-
can.
Well, Matt can, or he can at least make up some science for it.
I always
May or may not be real.
When drug companies talk about how great the vehicle is, I'm like, okay, your drug's not that good.
Yeah, I agree. But here's what's special about there. So it's that it has the surfactant is not a surfactant, is what is very unique about it. So they had to do special trials to be allowed to use this ingredient. It wasn't previously FDA approved for use in vehicles, and basically. With it, you can think of it as it's a solid up to 140 degrees. When it gets to 140 degrees, it melts, and then it's a surfactant. And so they make the product at 140 degrees to disperse it all. And then when they cool it all back down, it now sort of, you can picture it as like re-solidifying into tiny little specks, but those little specks are no longer a surfactant. So it's the first cream in history that you can legitimately say there's no surfactant in the cream, because surfactant, you have to have it in there to allow the oil and water to mix. Well, they mixed them at high temperature. Then whenever it came down to high temperature, now there's no surfactant in there. And so I also like this a lot for things that have it, like I'll use it especially in the face. If somebody's got dry, irritable skin, I think this is as a moisturizer would be probably be the most effective cream moisturizer we've ever had, because of this unique idea of heating it up, make it a surfactant, and then cool it back down. Now it's not a surfactant that's going to interrupt your skin lipids.
All right.
Yeah. No, and it should it looks like, they have these nice drawings of their little, micellar things, and now they, go into the lipid bilayer. So yeah, so it's interesting. So just a couple like quick interesting things. I was like, all right, is the foam really better? So I pulled the New England Journal phase 2 data and IGA with the cream, right? So same drug, but in a cream, not not a foam. So their IGA success was 31% versus 14% in vehicle. So that's like a delta, the spread between them of 17% on the body. If you look at the foam on this study, the delta is about 25%. So there actually was a bigger separation. But if you look at the scalp, it's like 38.6%. So it does look better. And then I was like, all right, how about like apremilast? They did scalp studies. Their phase 3B, if you look at, the patient, what percentage of patients were clear or almost clear with apremilast, it was 43% at week 16 versus 13% on placebo. So that is kind of like a delta of, again, 30%, which is actually not quite as high as it was for Zoryve Foam. That is my post hoc analysis comparing 2 trials, which you should never, ever do. Comparing 3.
Yeah. That's
Yeah.
good.
It's, Ferris, the biostatisticians are going to shut down UNC dermatology if you keep this up.
Exactly. But no, I
Shut
thought
it
it was
down.
interesting. So I've had very good luck with Zoryve Foam. So I am, it was good to see the data.
And Dr. Ferris,
And I
I
was
do.
in that study. So
I also have to give you the kudos that you did convince me of, like that Zoryve cream. It has a role, that the long term, we know the mechanism of action, we know it's safe, that there is a real it's got a meaningful place in atopic dermatitis therapy because of the long safety record that we've got with PDE4 inhibitors for little kids with atopic derm. So
okay,
thank you.
good. Hey, anything. I'm here to serve, Matt.
The servant leadership. That's right. That's how you got that
Exactly.
chair job.
Exactly.
All right, Patton, what's your last article here?
May 2025 issue of American Journal of Dermatopathology was titled Clinical Pathologic Features of PRAME Positive Common Melanocytic Nevi Case Control Study by Ronan Niddle et al. We talked about PRAME when Dr. Whitney High was our guest. So PRAME stands for Preferentially Expressed Antigen and Melanoma, which is actually PEAM, not PRAME, but let's move along. It's felt to be relatively sensitive and specific for melanoma. Dr. High said, like, in the last 10 years, what has been the biggest thing? And he mentioned PRAME when we were talking to him about that. So in the introduction, the authors, they all work in Australia. They mentioned that it's not infrequent to see otherwise benign lesions be stained for PRAME and then referred for second opinion because of PRAME positivity. So during a 1-month period, there were lesions coded as benign nevi with PRAME staining performed. And after some exclusions, they basically came away with 41 PRAME positive lesions, and they compared these to 43 PRAME negative lesions. They didn't really say how they selected the negative ones. I mean,
I got interested in that, so I dug into it.
Yeah.
I.e., used AI and fed the article in. They basically took 43 consecutive, just the first 43 that came in, 43 consecutive PRAME negative ones. Because that was like, how did they match?
Did they match them? Right.
They just said the first.
I read that, but like consecutive from the beginning, from the middle, around the just 43, they were negative. I mean, so the number of PRAME positive lesions, so it was like 800 some benign nevi.
Yeah, that got stained for PRAME.
Yeah, that got stained for PRAME. Only 62, so it was like 7-point-some%. And then they excluded partial ones, and then there was a melanoma in situ. They're like, that's not part of our study because that's not benign. And there was one that was a typo, whatever. So that's how they whittled it down to like that 41. I just used whittled twice in podcast. Yeah. Okay.
The first sign of senility.
So the lesions were examined by 3 dermpaths who didn't know the original diagnosis or the original PRAME staining, and they were scored as no atypia, mild atypia, severe atypia, and then scored on a 5-point scale for PRAME staining. The PRAME staining was not much to say there. It was pretty reproducible. Like what was called PRAME by the original like PRAME positive, it was also called PRAME positive. Like there weren't any discrepancy of that. So that's good. PRAME staining seems to be pretty reproducible and consistent. Compared to PRAME negative nevi, PRAME PRAME negative nevi. PRAME positive nevi more often demonstrated solar elastosis and lentiginous growth. That reached statistical significance. PRAME positive nevi more often had hypermelanosis, but this wasn't statistically significant more than PRAME negative nevi. Figure 1, I think, really was kind of impressive. It suggests PRAME staining seems to influence the pathologist's assessment of atypia. So if you look at the original diagnosis, where the pathologist knew what the PRAME staining was, way more lesions are classified as having severe atypia. So when the pathologist had no idea what the PRAME staining was, so the 3 dermatopathologists that kind of were reviewing these cases did not know what PRAME staining was. Only 3 of the PRAME positive nevi were classified as severe. The original dermatologist, like 22 of those PRAME positive nevi were stained as severe. So clear, like PRAME is influencing, seems to be, it's influencing the diagnosis of severe atypia. This does have clinical significance. Re-excision was performed on 44% of the PRAME positive nevi, read by the original dermatologist, compared to only 19% of the PRAME negative nevi. So yeah, that has to be, I would think, because PRAME positive nevi are diagnosed as having severe atypia.
That is very analogous to, so when I was a residency director, I got really into the research of like interviewing people, and it is the same thing. If you look at someone's CV and grades before you interview them, it dramatically influences your, if you then say, okay, now tell us how the interview was, don't think about their grades or their CV, just tell us how the interview was. If they had a great CV, you were like, if they were like a weirdo, you're more like, well, they're a little bit odd, but they, very smart, just blah, blah, blah. Whereas if they're like had bad grades and whatever, and they were a little bit odd, then you're more like, oh, they're just weird, they wouldn't be good, and the blah, 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 blah. So yeah, that pre-existing knowledge influences what is supposed to be an independent assessment. So it's kind of the exact same thing here. Pre-existing knowledge of PRAME makes people more likely to call atypia.
Yeah.
It's interesting.
It was weird. Like, these are tiny lesions, right? I mean, I think it's like 3 to 4 millimeter lesions. Do you guys biopsy 3 to 4 millimeter melanocytic? I mean, like, just as a general rule, I'm like, this is too small. I thought that was.
Rarely. I mean, if it looks really atypical, I mean, we've all had 3 or 4 millimeter diameter melanomas, but
I don't know if I have, because I've not been biopsying.
Well, there you go.
You've been missing them.
They came to me and I biopsied them.
They got a second opinion.
No, I agree. Like, I'm much more thoughtful about, size as a factor for considering that. But yeah, I think, it's like you really need clinical pathologic correlation. Like, PRAME is not the magic bullet. So it's like you need your kind of pretest probability, right? So if this is a 3 millimeter nevus that the pathologist thinks looks benign, and then you go stain it with PRAME, you're like, don't even go stain it with PRAME, right? It's like when we talked about GEP. If it's a 0.2 millimeter, 0.3 millimeter melanoma, don't get GEP. The pretest probability is so low that is going to be a metastatic melanoma that, you don't have, the predictive value of the test becomes very poor, right? Pretest probability is key.
It seemed like they were willy-nilly, like staining everything with PRAME. They had like 800 lesions in 1 month that they stained with PRAME.
Yeah, and so the other take would be like, call your atypia before you know what the PRAME is. Like, that seems to be the lesson here. And maybe for severe atypia that is PRAME positive, maybe those are the real, incipient. What's the report we get? May represent an early evolving melanoma in situ. Like, maybe the PRAME positive severe atypia, where you classify severity before you even know what PRAME is, maybe those are the real melanoma precursor lesions. I don't know.
Yeah,
I'd like Dermpass to be more honest in their reports and say, may represent a lesion I could get sued over, so you should re-excise it.
I'm always so grateful that I'm not the one who has to make that final call. I respect our Dermpass. That would be so hard to have to make that call.
I thought about doing Dermpath, and the reason I didn't was because I didn't want to. I literally didn't do a Dermpath fellowship because I didn't want to have to do that.
Yeah.
That was before PRAME. Now with PRAME, maybe you should reconsider that.
Right.
There's a ton of pharma money in DermPath. You'd fit right in.
Big
Okay.
PRAME. Big PRAME's out to get you.
All right. Well, those were some good articles, a good clinically useful 6-pack. And, any final comments for the week here, Dr. Patton or Dr. Ferris?
I'm good.
No.
Okay.
Saving lives.
Saving lives, right? Skin
Doing it.
cancer.
That's right.
Exactly. Who was somebody? I was talking to somebody recently who didn't know that there was a TV show called Pimple Popper MD. Whenever I brought up, Pimple Popper was there, they were like, Seinfeld was there.
Oh gosh.
Were
They've
they a
been living under a
dermatologist?
rock. Everybody. If I have a clinic day where nobody mentions Pimple Popper,
Yeah,
it's
that's
an
very
anomaly.
surprising to me.
Yeah, it was bizarre. It was hard to believe. All right, I want to thank everybody for joining us today. If you got questions, comments, ideas for topics we should cover on the show, shoot us an email at dermsondrugs.com. We hope you learned a few things. We hope you laughed once or twice, and mostly we're hoping you're planning to join us next week. And until then, I'm Matt Cyrus.
I'm Tim Patton.
And I'm Laura Ferris, and we are Derms on Drugs.